All right, get started. All right, guys. So our review is going to be for the first three topics. We will be having review sessions tomorrow and Sunday. Also, time and location to be determined by the TAs that are running those sessions. But yeah, so also important to clarify, we're not going to be able to go over every single little piece of detail about all the topics. So do not heavily rely on this being the only information for these topics. We ask that you guys take a peek at, all the notes, everything you've been working on so far. So brief introduction about amino acids. So amino acid structure, you're gonna have central carbon, you're gonna have the amino side, you're gonna have the acid side, which is the carboxylic acid ending, and then you're going to have an R group. And the R group is what's going to be characteristic of the specific amino acid we're talking about. It's been a while, I know, and that's just simple amino acid structure. So here we're going to be talking about the formation of the peptide bond. So the formation of a peptide bond is a dehydration reaction. What that means is that you get, along with the two newly joined peptides, you'll be getting a water molecule out of this reaction. This bond is characteristic of the backbone structure of a protein, and it is a very interesting covalent bond because it shares double bonds, or it has double bond characteristics, though it is a single bond. And more on that later. And characteristic of this specific type of bond, also, you need a hydration reaction to occur for you to break it. So any sort of enzyme that is breaking peptide bonds for any different proteins that we might have looked at will be having a hydration reaction. So the different levels of protein structure. Oh, maybe I should go back and talk about that bond. Okay, going back to the double bond structure of that peptide bond, because I thought there was going to be more about it. It is a smaller in length bond than your average covalent bond. What that means is that it'll have stronger nature than a regular, bond. And so also what's very characteristic about this specific bond is that because you have all those electrons coming from either being shared from oxygen and nitrogen, you have the strength of, what's the word? It's so embarrassing. Yes, resonance. Sorry, guys, resonance, which is why that bond continues to be strong. Okay, so let's move along to the protein structure levels. So we have primary structure. Primary is the sequence of amino acids of a protein. And peptide bonds are the ones that are going to be holding it together. And this process requires a lot of energy, and you can only get that done via enzymatic activity. So primary structure, just the sequence of amino acids. Any questions so far? No? Okay. So secondary structure. So here's where it gets a little bit more interesting. And it is reliant on the interaction between bonds of the backbone, and it is reliant on backbone atoms. So this is not R group interaction. Very important to remember. If you get asked a question about alpha helices or beta sheets, do not talk about the interactions between the R groups. These are just going to be the oxygens and hydrogens present on the backbone atoms. So you are not reliant on R groups. And how does this happen, John? Well, you have the presence of lots of hydrogen bonds, which is how the structure of these specific secondary structures will be held together. And these interactions are non-covalent, and this is why we are heavily reliant on the conditions being optimal for the protein to be stable, because these are hydrogen bonds. And so you are not going to have any sort of structure being held together by the other covalent bonds. Oh, also important to mention the structure for these, for the helices and for the sheets. For alpha helices, the R groups are going to be sticking away from the sheet or from the helix. And for beta sheets, that is going to be it goes back and forth. They're alternating on the sheet. And as you can tell, well, it's actually hard to see. There's arrows on the beta sheet. And usually on a secondary or like on a on a ribbon structure protein, you'll be having them drawn as like those little like grooved arrows. And the way you can determine which way it's going, you just have to look at where that swivel is going to be around. And so you are able to tell that the R group interaction or the backbone atom interactions are going to be holding them together. And on one side of them, the R groups are going to be sticking up. The other side, they're going to be sticking down. So they're not going to be close to one another. And that's also important for things like permeability and pores that we'll be talking about in a second. Any questions? Yes, Mario. No, but
what do you mean by ubiquitous when you say backbone interactions make alpha helices and beta sheets ubiquitous?
do we? I don't.
Does that mean that they're just like present everywhere? Is that what you mean? Or
Yeah, I think. Well, that probably goes along with the prevalence of these structures and proteins.
okay,
I'm not exactly shabby as these slides are kind of but they're very prominent as structures for all sorts of protein interactions and just protein structure in general. But not something like we're not gonna. The question won't be like, oh, like how ubiquitous is this sort of level structure? Like, it's just about the prominence, and they're just very important because of like the interactions you're able to have, because of the way the R groups are structured. And because of the way that the different helices exist, the different, we've seen pores made of beta barrels before. So just the types of interactions that can happen because of that. Think about where the hydrophobic amino acids would be located. Think about where the hydrophilic amino acids would be located, and just things like that. Okay, tertiary structure. So this has to do with the overall protein shape. And this is gonna have to do with the interactions between the different R groups that are present. And so you're gonna have a whole variety of different interactions depending on where the amino acids are located within the protein structure. And most of these R group interactions are going to be non-covalent, besides one very, very important structure here that we have, which is the disulfide bond. Anybody remember which amino acid is involved in that? Yep. Yep. And why?
because of
Yeah. And so you are having these very strong sulfide bonds that are holding these proteins together, or these proteins together. If you do work in lab with PCR, you'll know the existence about Taq polymerase. Taq polymerase is a polymerase that comes from a specific species that lives in thermal waters. And I like using this example that Rosella used when I was a student. Just these thermophilics are the names of the animals that live, or the species that live in like these very warm temperatures, and their protein structures are filled with these because they're covalent. They're able to take on more heat variation and just stabilize the proteins. Any questions? The disulfide bonds are covalent. They're the only tertiary covalent bonds. Oh, I should probably talk about the different bond types. So we have, okay, what are the different bond types, guys? Okay. Okay. Yep. We're missing one. That's the same thing. Well, we're missing two then. Dipole, and what else? Yep. And what type of bonds are hydrogen bonds specifically? Dipole. Okay. And then quaternary structure. So this is when you have the interaction between two different polypeptides. And the interaction that we're going to go over is just how those proteins are being lodged together. Yes.
The types of bonds, did you go through like them ordered in strength? Because I know that like showed up on the columns.
So it goes dipole. Well, it goes dispersion forces at the least, and then it goes dipole, ion dipole, ionic, covalent. Hydrogen bonds are dipole bonds.
Can you repeat that?
So it goes van der Waals, dipole / hydrogen. Ion dipole. Ionic. Covalent.
And Van der Waals would just be the same as, hydrogen.
Dispersion.
And is electrostatic the same as ionic?
You mentioned ion bonds are the most intense, right?
They're the second strongest.
I just wanted to.
Okay. So. Yeah,
Do the non-covalent bonds besides hydrogen bonds show up anywhere besides tertiary and quaternary structures?
like in general.
Like in protein folding?
Well, they appear in secondary structure for maintaining the structures of the helices and sheets and with protein folding with the different R group interactions.
How do they show up in secondary structure?
The backbone atoms.
Okay.
Any other questions? No? Okay. So many subunits together can form a very large protein. Hemoglobin's a good example, insulin's a good example. There's plenty of different proteins that are made up of different subunits. And though very important, tertiary and quaternary structure are not the same thing. And though every subunit exists in its tertiary structure, not every single protein will have quaternary. Yes? In secondary structure, they're just they can be hydrogen bonds or they can be dipole bonds.
For us to know, like in, like there's like 4 amino acids today in the alpha glucose. I just remember that being,
That was an example question, right?
it was a, problem
Problem
set
set.
question.
That was a problem set question.
Yeah.
Yeah, so she can definitely throw that type of question in there. I think because of the way that the hydrogen bonds happen and because of the way that the helix structure happens, you need at least 4 amino acids to start turning. Because if you think about how 1's gonna bind with 4, 2 is gonna bind with 5, 3 is gonna bind with 6, you want to have a consistent amount of like R groups or not R groups, backbone atoms so that they can actually form the structure and hold it together. Questions? Okay.
I remember specifically in the quaternary structure, there were 2 confusing names and stuff, like ions and stuff, but.
Yep, we're gonna get to that in a second. Oh, yeah. Domain and subunits, there's a slide on that. Yep, okay, so just a little review. Tertiary structure is a single polypeptide chain and how it folds. Quaternary is multiple polypeptide chains and how they fold in the 3D shape. And like we just talked about, there's different types of atom interactions that will hold protein structures together, going from the way the protein folds, the way the protein interacts with other proteins. Okay, domain subunits. The domains are independently folded portions of subunits. And that just goes to say that there is a specific portion of a specific protein that will fold in a certain type of way. Domains are highly conserved evolutionarily, so you'll have a whole bunch of different domains for different proteins that are kept the same throughout like a whole variety of different species. And it just goes to show how important the function of these specific domains are. I remember for one of the, was it the problem set that had the structure of an antibody? Yeah, so it just goes to show, think about how the immune system works and how important the different domains for the specific antibodies are. And so, Sabine, you want to talk? You want to say it? You can say it.
The only subunits are 2 different glycopeptide chains coming together. You only see quaternary structure when you have 2 or more different like chains of amino acids.
Yeah,
Can you subunits only happen in quaternary structure?
Go ahead.
Yeah.
So domains independently folded, you can have multiple different domains in a single subunit. So think about,
This in tertiary structure all the time.
yes. So it's just, yeah,
I'm curious, like, so if you, because I know that she's talked about like Chinese domains and, you know,
Mhm.
what have you. But could you also have like any subunits?
so a subunit is just a protein.
Right,
A specific protein will have different types of functions, and each of those functions is genuinely, not every single time, but generally speaking, every single function performed by a different protein will have its own specific domain. So we were just talking about DNA replication in class. The polymerase has polymerizing domain and the exonuclease domain, right? So different functions are typically performed by different domains. Any other questions? Yeah.
In a model, is there like an easy way to identify a domain?
Just, it's usually reliant on you being able to see the close interaction and the way a protein, because usually like a domain will like stick out away from like the other parts, because the protein folds in a specific way to have the domain exist. There isn't really like a determining way to
So I
see
guess
how
like
that.
within one subunit, a part that's like hanging out, holding its own.
Yeah, not, well, it's not a subunit.
I mean, within a subunit.
Within a subunit, yeah. The way I like to think about it is, you could think about a protein as a train, and each of the specific wagons will be a domain. So just how I like to see it. Okay. So we can move along. Oh crap. So our group characteristics, interactions and bonds. I believe you are provided the amino acid chart during the final. But I would just be familiar with the way that the chart is set so that you don't really have to spend that much time thinking about how it works. You just kind of want to stay away from like not looking at it at all beforehand. Sometimes you can get thrown with some different types of questions, but looking at it, polar charged star groups are, they're very hydrophilic, and so you will be able to see them in aqueous solutions. They won't be hiding away from the water. And so you have the polar charged, or the positive charged and the negatively charged polar amino acids. They are, all of those, besides the non-polar ones, are polar amino acids. Those 5 just happen to be charged, while these 5 are not charged. And so polar uncharged groups, which are the blue ones here, will be also very hydrophilic. And they'll be having a difference in electronegativity. But the biggest takeaway from the difference between those is that those will have a charge. These can interact with the charge in ion dipole, but they will not be making ionic bonds. And the nonpolar R groups, they're typically, not all of them, but you'll see the bulkier groups tend to be nonpolar. You have tryptophan, you have phenylalanine, which are both very bulky, very cyclic. And then also I want to point out a couple of the nonpolar ones that have special features. You'll see glycine a little bit, and you can be asked during the exam, oh, like why is glycine an example of a polar or a non-polar amino acid that can be considered a little bit more polar? That's just because it has a hydrogen, it's less bulky, it fits into different spaces, and it we'll go over the electronegativity chart in a second, but it has just more room to be able to interact with that sort of environment. And also proline, special one, it is a helix breaker. That's because the carbon groups are making an the carbon group is making an interaction with the backbone nitrogen, and it is forming a cyclic. It's supposed to be, it's a pentagon, right? But it's not being able to form the different secondary structures because the electrons are not able to be shared while in a cyclic configuration. And so if you see a ribbon structure being drawn, you'll see that the helix usually ends and then there's a little loop. That little loop is usually a region that's rich in prolines. Any questions? Yes. Both the prolines?
Yeah.
Yeah, so the prolines can't make backbone interactions, which is what holds secondary structure together. Which is why, in the ribbon structure drawing, like you'll see, I don't know if we have a couple back, the next slides. But, like in a ribbon structure, you'll see that they usually break at the end and then they go back into a ribbon structure again. Or a helix structure. That region of the break of the helix is usually rich in prolines because they can't make the ribbon structure. Yes,
I may not have heard it when you were explaining it, but is it glycine and alanine that can act as both a polar or non-polar molecule? Or I can't remember. I remember there being one amino acid.
yeah, so glycine is. I mean, you can defend the point of view that it is a more polar, non-polar amino acid, as counterintuitive as that sounds, because of the electronegativity of hydrogen, because of the small structure, it can fit into different spaces. And it can just interact freely with, more. If there's a dipole, though the non-polar amino acids are theoretically not supposed to, like, it can interact with that sort of dynamic.
But alanine can't, it's just glycine that does that?
No, alanine is a little bit bulkier, like the CH3, the way it's drawn doesn't really look bulky, but if you think about the way that the hydrogens can behave, it is a little bit bulkier because of the methyl group. But it's still in comparison to literally every single other amino acid, you can have those interactions happen more frequently. Yes?
How come you can't make the backbone interaction?
So the backbone interactions happen within the central atoms of the backbone. So the central carbon and nitrogen, those are the two molecules that can make those interactions, and the oxygen on the carboxylic acid. So because the nitrogen is stuck with 4 atoms, it has a plus sign. And it can't make that interaction no more because of the cyclic nature. So there's no escape route for the electrons to be shared elsewhere. So you don't have the resonance. Yes.
2 questions.
Mhm.
First of all, when you mean similar electronegativity, do you mean that they're all around the same level of electrical charge to each other? Because I'm noticing that there are 2 groups, one that has a positive charge and one that has a negative charge.
So they have different, so electronegativity is a measure of how, I guess you could say, quote unquote, and do not quote me on this, hungry for electrons the atoms are. And so a way of just measuring that is looking at, if you look at the periodic table, fluorine is the most electronegative atom. And that is just because it holds on to different, to a higher level of electrons than a molecule like carbon would. If you picture the drawing of what water looks like, typically the oxygen atom, if you look at like a heat version of a water molecule, will have higher electronegativity around the O instead of the H. But that's because the oxygen in nature has more affinity for the electrons.
Oh, gotcha. You're saying that it's not polar. Basically, that's what I'm getting. It's polar.
No, water's polar.
I mean, yeah, wait, they are polar? Or wait.
Water?
Oh. Okay, okay. Okay, sorry about that.
No, it's fine.
I just had a brain fart. Okay, my second thing.
Okay.
Is how do you define van der Waals? How can we tell if it's van der Waals? You mentioned covalent bonds for non-polar R groups, we also mentioned one that is for negate van der Waals.
So van der Waals interactions will happen when nonpolar R groups come in close proximity to each other. And so there's these things called Brownian motions that are just how the electrons and the atoms are just floating in space, right? And so there isn't really anything pushing them or pulling them, but these forces that I just mentioned, and they just happen to move the R group in a direction that's towards another R group. And because they can't interact and there's no enzyme to make a covalent bond between those 2 atoms, they just move along and hold close to each other because of the similar hydrophobicity.
Gotcha. Yeah, that seems right. That's what.
Mhm.
And
So, yeah.
covalent bonds, though, I'm assuming it's like two bonds with the exact opposite charge, plus the other and perfectly balance each other out.
Covalent bonds?
Either that or it's ionic.
Ionic bonds between two different or two opposite charged atoms, yes.
Okay, that's, yeah, covalent though. We might actually have to describe it.
A covalent bond?
Yeah.
A covalent bond is a bond between two atoms where the electrons are being shared.
For the electron. That's usually extremely similar to ionic bond. I'm guessing. Because ionic bonds, like, they don't have opposite charges on the kind of like.
Random bonds, are totally different, yeah. Even though like it's different.
That. Seems like it is different a little bit, like it is there, but I don't classify it as almost the same thing.
So covalent bonds are, at least in this aspect of class where we're not looking at inorganic chemistry, we're going to say that covalent bonds are stronger. Okay. Moving along, so this is the hydrophobicity chart that I was talking about earlier. If you want to write this down for one of your cheat sheets, you can. It's a good chart. It seems a little bit counterintuitive because of the way you look at the different amino acids in the ranking. But the biggest takeaway is that you're going to have your polar and a couple of your charged amino acids being more hydrophilic than, let's say, your isoleucine. And that's just because of the way polar molecules can interact with each other and the way that the protein structure will lead these different amino acids to being located in the protein. So highly unlikely for you to find something like a phenylalanine on the outside of a protein because it just has way too many different approaches for reactive oxygen species or any sort of other highly reactive molecule to accrue it. And so you also want to think about how these different structures will position themselves together in a situation like oil trying to mix with water. So if you ever mix oil with water while cooking pasta, as you should while cooking pasta, you'll see that you form little bubbles of oil, and that's just because the nonpolar oil is in a aqueous environment and it is just positioning itself in a way so that it doesn't mix with water because of the hydrophobicity of the lipid tails. Good. Okay. And this is just again, it's just to reiterate how important the disulfide bonds are. This only happens in protein structure. I mean, it's the only covalent bond present in tertiary structure. And you need two cysteines. So this is just a little bond summary. I know we went over this a couple slides ago. But primary structure, secondary structure, tertiary and quaternary. Those are it. Something that I'll say, sometimes students forget, if like there's a question that asks if you're modifying a polypeptide chain and changing one nonpolar amino acid with the other, they get asked what's going to change. I think this might have been a question on one of the exams.
Yeah.
If it asks what it's going to change and you only talk about how the hydrophobicity will remain the same, you were partially getting the question wrong because we're changing primary structure. So just for every different modification that's happening, think about the protein structure that can be affected. If you're changing it at all, if it's not the same as the original, you are changing primary structure. Now, if you're being asked if it's going to denature, if it's going to change protein shape, if the shape's going to change the function, yes to all of those. But if you have a modification on the chain, on the way that the protein is in primary structure, then you will be having that modification. Yes.
I know, like, I saw this marked on some, like, on my exam for exam one.
Mhm.
How much, like, if you're replacing, let's say, aspartic acid with glutamic acid, since they're both, like, very similar structure and both same negative charge,
Mhm.
does the difference in size between them, since it's not major, have a impact?
It does because you can have a modification on how the protein will position itself. And let's say if it's the binding pocket of an enzyme substrate interaction, if you change and it makes the affinity higher, then, or if it changes to, let's say, glutamic acid being smaller, then you can have higher affinity because the protein is able to lodge itself better into the substrate site.
Also, follow up to that with like the binding pocket questions, because I know that was on the wrong side of the exam.
Mhm.
With those types of questions, if you're replacing an amino acid with another one, is it always gonna make the bond that's the strongest? Because like the question would be asking what kind of bonds or what kind of bonds would you see forming? Is it always gonna default to whatever the strongest possibility is?
Yes.
Okay.
So let's say you change from a negatively charged amino acid to like a threonine, or I don't know, like another, like a tyrosine. You're going to default to, you have to think about the possibility in bonds that can happen. So don't just go from saying, oh, you're gonna move from, it's not, I mean, they're both polar, so I'm still gonna have polar non-covalent interactions. If you're moving from an ionic bond to an ion dipole, that bond's weak. So you gotta think about that.
Yeah.
Yes?
I was actually going to ask you this, I remember looking at the answer sheet for the exams, and yes, if I did, we will have an impact, maybe we'll take points, but that we don't.
Okay, so question for you guys. Which of the following amino acids be bound to an amino acid with a positively charged R group and a tertiary structure? Anybody? I'll call on people.
Glutamic acid.
Glutamic acid. What else?
Yeah, I have a question.
Yeah.
What frequency is?
Oh, we're not doing clicker. Just shout the answer out.
Oh,
No, we're not doing clickers.
yeah, if I can actually use it once, that's third.
Anyone else? Okay. So you are correct. Anything else? we wanna ask? No? Okay.
In terms, I can wanna.
Oh.
Thank you.
Mhm. Alpha helices and or beta sheets are found in every protein. What characteristic of their bonding makes this possible?
They use the backbone structures in an amino acid, and that's consistent through all of them. Besides, I guess, proline, but
And what's a way to disrupt those?
um,
What's a couple ways to disrupt those?
there are like, depending on like temperature, pH, salinity,
Mhm.
light, things like that can denature and cause misfolding.
Okay, good. Okay. And then just the different ways to model proteins. This goes for different ways to portray and to focus on what you want to focus. If you are dealing with more of how the domains might look, then you have, usually the ribbon structure is the best for that. There's space fill. There's backbone, which is, I think backbone's pretty lame, I'm not gonna lie. But there's backbone if you're interested in that, and you don't show the R groups, and then there is ball and stick, which you show the atoms and the R groups.
Maybe some more ways of structuring protein stuff.
Okay. Oh, question? Yeah. Do you want the answer? So the hydrogen bonds that happen between the backbone atoms are what's what makes it possible for these interactions to happen. And the way to think about that is that you'll have backbone atoms in every single protein. And that's why you're able to have these evolutionarily conserved structures.
Are the polar molecules still able to make covalent bonds with non-polar molecules, or do they just not interact at all?
I would like stay away from like trying to write. I don't think there's, I mean, in nature, yeah, but for this class purposes, like you don't have to.
Okay, so it's really only just gonna be polar molecules interacting with polar and then non-polar with non-polar.
Well, she might ask you like how some sort of protein would interact in like a non-aqueous environment
Right.
or aqueous environment. That's like as far as that probably would go. Yeah. Questions? Okay. Go ahead, Jen.
Okay, now we're gonna start topic 2. We're gonna start with protein unfolding and misfolding. So the main ideas of this topic is that the environment that a protein is in is going to impact its folding and its functions. Some of the environmental factors that could lead to denaturation is temperature, so heat, pH and salt concentrations, and light. A cell is able to regulate all of these factors just because it wants to maintain, stable conditions. It wants all its proteins to regulate normally. And when a protein misfolds or unfolds, this is not normal and the cell needs to get rid of it. The unfolding of a protein is called denaturing, and renaturing is when it refolds. And like I said, cells don't want misfolded or unfolded proteins. Think about how structure affects function. If structure is messed up, you want to get rid of it. So how are the bonds affected through these environmental factors? So all of these factors are most likely going to impact your non-covalent bonds. And that's because, as John talked about in this class, we consider our covalent bonds to be the strongest. So these environmental factors are going to disrupt your non-covalent bonds, which includes all the bonds that we've been talking about, your hydrophobic interactions, your ionic bonds, and your hydrogen bonds. And for covalent bonds to be broken, you're going to need intense, very, very, very intense heat or very, very, very intense light. So most of the time, we're not talking about the disruption of the covalent bonds, we're talking about the disruption of other bonds. Okay, so now we're going to talk about heat and how heat denatures proteins. So it attacks non-covalent bonds and causes the unfolding of a protein. Like we talked about a few seconds ago, hydrogen bonds are going to hold your backbone together. If you're disrupting those hydrogen bonds, your protein is going to unfold and it's not going to maintain its structure. In general, the cell wants to maintain a Goldilocks state. It wants to be in its just right state in terms of heat and pH and salt concentrations. Different parts of our bodies have different pHs. Our proteins need to maintain just the right temperature, light, pH, and salt concentrations. Higher temperature or lower temperature doesn't always mean that the function of the protein is going to improve or if it's going to be worse. You kind of, like I said, Dr. Francis talked about this. The cells need to be just right so that they can bind and unbind so ligands can bind and unbind and that they can execute their functions. And then pH and salt concentrations, these can affect your ionic bonds just because the charges that are present in the cell can be broken and then want to bind with something else, which will disrupt your ionic bonds. And then light can also be introduced to a system and disrupt those non-covalent bonds that hold the protein together. Like I said before, very, very intense heat and very, very, very intense light can break covalent bonds, but generally we're talking about the disruption of non-covalent bonds. So so like light wavelength. So how does the cell go about fixing these problems? We have a few different responses that you're responsible for knowing. So the heat shock response in the cytosol nucleus, and then the unfolded protein response for the proteins that go into the endomembrane system. And then some neurodegenerative diseases are caused by misfolding proteins, mad cow disease. I think like glaucoma has something to do with aggregate proteins in the eye. And then these aggregate clumps of proteins that have denatured and like can't refold are insoluble. And the hydrophobic core becomes exposed.
So to help,
So it says that, for the heat shock response they're sent to proteasome, is there any like, function that tries to repair the misfolding before being sent to the proteasome?
so there are chaperone proteins. I'm gonna talk a little bit more about the heat shock response in a second, but basically I'm gonna cover it in just a second, but chaperone proteins are kind of like that first response. They will sense the misfold or the unfold, and they will help a protein refold back into its natural state. I like to think that these proteins are like chaperones on a field trip, like gathering up little kids and making sure that they're doing the right thing and that their structure is good and that their function is good. So in this little like chart, you can see, so a protein is made, and if it's unfolded, and it can't be refolded by the chaperone, it'll just aggregate because you don't want a protein that has no like valid function. But if the chaperone is able to refold it, it'll be folded and it'll be good to use. And then the response, yeah.
Is there a reason the chaperone proteins don't like, in a heat shock situation where the cell is at a higher temperature, is there a reason the chaperone proteins don't misfold?
I think like their structure and their function is likely made to be able to like endure higher heats. So it's probably just related to its structure and its function. I know we didn't talk a lot about like the mechanism of it, but it's just like likely evolutionarily conserved to be able to endure higher heat.
So why does chaperone actually get upregulated if it's going to get destroyed?
What's getting destroyed? Sorry.
It says that. Aren't chaperone proteins like helping them like trying to hold back together?
Yeah, so in the case of unfolded protein response and heat shock response, you're gonna have an upregulation in chaperone proteins, so that like, I know this chart is a little confusing, but if it's unfolded, the chaperone protein, you need more chaperone proteins because you want more of them to get refolded. You don't want like unfolded proteins to aggregate, so you need more chaperone proteins to help refold. Usually chaperone proteins are good about refolding proteins. That's why they get upregulated.
Do chaperone proteins also chaperone misfolded proteins to the proteasome, or is that a different protein that would? Or is that like, do they get ubiquitinated?
They get tagged, yeah. Yeah, to the proteasome, yeah, and then something else sticks into the proteasome.
Got it.
So heat shock response is the response that happens in the cytosol in the nucleus. The cell is going to sense that there is a protein that's misfolded, something is wrong, like something isn't working as it should be. And so chaperone production is upregulated. Again, you just want the chaperone proteins around to help the protein refold back to how it has to be, to its natural state. And then the protein is tagged with ubiquitin, and then it is sent to the proteasome to be degraded through ubiquitin-mediated degradation or ubiquitin-mediated proteolysis. Yeah, so the proteasome kind of acts like a garbage disposal for these proteins.
This is, I guess, kind of jumping ahead with other processes, but like, I know with nuclear import, the proteins have to enter unfolded. How does that work with chaperone proteins, for them being imported into the nucleus? Do they follow that same process? Is that
Do
even
chaperone
relevant?
proteins?
Should I even-
No, the nuclear protein already folded.
They're not. Oh, they are. No, they're not.
Aren't the Golgi and the ER protein unfolded and they folded?
Oh, sorry, it might be, okay.
Yeah.
Maybe.
The nuclear
You might be right.
protein is already folded.
Okay, they go in fully folded.
We'll also cover that for topic 3.
We'll be there soon.
I don't know if it's all covered in topic 3, but so like, do the misfolded proteins exit the nucleus in their misfolded state? And do they have to have a nuclear export sequence to use them?
I would assume so. I would assume they have to have a nuclear export sequence because they're leaving the nucleus and they're going to the proteasome. Or, yeah. Yeah, maybe. But there's no proteasome in the nucleus.
Yeah, I mean,
So they have to leave
if
the
they're
nucleus.
supposed to stay in the nucleus forever, then there's probably specialized chaperones
To get them out.
get them back. But if a protein only has an import, no export, then it typically just stays.
Right.
Yeah.
So if it was meant to be degraded, it would probably be tagged or otherwise somehow help to leave the nucleus so it can be degraded or.
Do the chaperone proteins follow the protein that it's bound to into the proteasome and get, degraded as well, or is it released and then tagged and then the protein is sent?
Chaperone proteins shouldn't be degraded along with the protein that's getting degraded, from my knowledge. Yeah, they shouldn't be. Unfolded protein response. So this is the response that happens in the endomembranes in the ER. So a few different things are gonna happen to kind of deal with a misfold or an unfold. You're gonna have translation decreasing in the ER. This is because you don't want to make new proteins while you have a protein that's unfolded and non-functional. You want to fix the problem at hand before you upregulate regular proteins. You're also gonna have the increase or the upregulation of chaperone proteins. Again, chaperone proteins are there to kind of fix the problem. You want more of them around, you want a higher concentration of them. And then lipid synthesis is going to increase just to maximize the size and the volume of the AR to kind of accommodate the increase in chaperone proteins around and to increase the volume. And then most importantly, there's no proteasome in the ER, so the protein must be retrotranslocated out of the ER so that it can be sent to the proteasome and then be degraded.
Can you explain how lipids are used to increase the size of the ER? But does that even matter?
I think lipid. That's a. I just know that it does.
Okay, that's good enough.
Let me, John, do you know how lipid synthesis increases the volume of the ER? Or does it, that's it, it just does?
It just does.
Yeah, okay.
Is she gonna expect to say emotional feelings on the exam?
As long as you
Yeah.
get your point across.
Like if you know, yeah,
Yeah, you don't have to use the exact like verbiage, just explain, kind of, if you can explain in different terms what that word means, then.
yeah.
It means something.
You could either use the word or the definition of the word.
I think something basically the same language, yeah. It's like the same exact thing.
She didn't create the example.
Oh, yeah, or, yeah, I can hear it. I don't know.
How come if you're decreasing overall translation, how are you increasing translation of chaperone proteins?
So from what I know, the ribosome, you're gonna decrease the translation of regular proteins that are folding in the ER, but yes, the cell knows. Or there might be like increased translation of chaperone genes, and therefore you're gonna have upregulation, just because if there's a higher concentration of mRNA to be translated, there should be an upregulation in the translation of chaperone proteins. But you're not like upregulating regular proteins, so you're able to decrease the production of those proteins. That's
It's not
how I
be-
understand it.
It's not because you're translating or you're downregulating something that you have to downregulate. Like, if you think about the way that the like bacterial genome or the human genome is, the genes aren't like juxtaposed, they're not on top of each other. So you're able to overly express something at one particular part of the genome while under expressing another. So if you think about how heterochromatin works, how euchromatin works, and how that allows or prevents you from getting to the specific genome regions, that can like help you visualize how the translation will work.
So, any questions on that before we move on to protein ligand interactions? Okay, so this is kind of a summary of everything I'm going to talk about. So allostery, it's just the concept of how protein function is regulated by the structure of the protein. And then post-translational modifications, the chemical modification of proteins after that they've been translated and made and that they exist. Some examples ubiquitin tags, acylation, and the adding of phosphate groups, phosphorylation. And then nucleotide binding and hydrolysis. So the regulation of proteins through the binding and hydrolysis of either ATP or GTP, and then the hydrolysis of those molecules. And the hydrolysis is always leaving the bond between the second and the third phosphate. Always. Okay, so allosteric, this is basically just regulating a protein through something that binds to anything other than the active site of the protein. So here you see that it can act as either an activator or an inhibitor. So something can bind to like somewhere that isn't the active site of the protein and cause activation. So it allows for the ligand to bind to that active site. So that's acting as an activator. And then there's inhibition, where something will bind to somewhere that's not the active site, and it'll inhibit a ligand from binding. And that's just a mechanism or a way in which proteins can be regulated through their structure. And then post-translational modifications usually happens at a specific amino acid. And these are some examples that we even talked about in later topics. So phosphorylation, adding of phosphate groups, just their insights, or tagging the protein with ubiquitin so that they could be sent to the proteasome and get degraded, like we just talked about in unfolded version. Yeah.
Can you explain like what's the again?
Yeah. Allosteric is just like structure affecting function. You want me to like talk about all of it again?
Just like, so I get that like it's regulating it by like you said anything but the active site.
Mhm.
So what is the point of that? Like why can't it go to the active site to regulate it?
It's not gonna go to the active site. Like for activation, for example, if it went to the active site, then your ligand can't bind at that active site and it serves no purpose. So that's why it binds somewhere that isn't the active site, because it doesn't like, it's not in theory like interacting to cause a response, or I mean, it is causing a response, but if it were to bind to the active site, you wouldn't be able to have that ligand binding there. And it's just the way that it works. And then in like the inhibition, it kind of closes that active site or just inhibits the ligand from binding at that site. So that's why it won't bind at the active site. And that's,
Does it like allow the ligand to bind there?
yeah, exactly. So in activation, it would allow the ligand to bind at that active site, but if it's inhibiting, it would prevent the ligand. Any questions about post-translational? Okay. And then nucleotide binding and hydrolysis. So ATPases and GTPases are enzymes because they bind to ATP or GTP, and then they hydrolyze that bond between the second and third phosphate. They cleave that bond. Yeah, it goes from a triphosphate, so ATP, to a diphosphate, ADP. And then we've seen this in like the signaling pathways. We talk about this like later on in the class. So just it's important to know that this is a method of protein regulation. Okay, now, yes.
So one form of regulation would like competitive inhibition be, or is that would that be something separate from all three of those?
I would say that's allosteric. But hold on, let me think about it for a second. Because it's not the active site. What? I guess it would be different then, it's its own like. would say it's its own. It's none of these.
Yeah.
Yeah, it's its own, like, little niche.
I mean, it's still continuation of protein activity because you are competitively inhibited, which means like, if you need ATP molecule to bind and you have a phosphomagnetic or a protein that will inhibit the ATP binding because it binds directly at the active site, like it's still protein function inhibition. But it's not like, yeah,
It's like its own little niche.
it is.
Yeah, it's its own like. It's not exactly allosteric because it's binding at the active site. But. But yeah, that was a good question. Anything else? Okay, KDs. Take home message for KDs is the lower the KD, the higher the affinity. KD is a dissociation constant. It's how often a protein is going to dissociate from its ligand. The definition of a KD is the concentration of ligand when protein and protein complex with ligand are equal. I wouldn't like think too deeply about that. I would know what it is, but just remember that the lower the KD, the higher your affinity. It's just a measurement of the interaction between a protein and a ligand. Any questions about KD?
What's like, oh, son of what was like that question, on the problems at our exam that was like, with like the substrate versus product with KD, I can't remember exactly what it was, but like or the enzyme wants or whatever, like higher, is that
Yeah.
higher KD for the product?
So it should have a higher KD. Yeah, no. The higher the KD, the lower the affinity for the product. Yes.
Okay.
Yes.
Thank you.
The mechanistic way to think about that is the more the enzyme's holding onto the product, the longer it takes for it to be able to get to a substrate.
Okay, that makes sense.
Yeah.
And then enzymes. Okay. Cells usually need enzymes to make and break covalent bonds. It's just like a time thing. It makes it easier, faster to make and break these covalent bonds. And then enzymes are regulated. Ribozymes are enzymes that are made up of protein and RNA. And RNA is the catalytic component. Yes.
So for the slide before that.
Yeah.
Would there be a
Wait.
case where like. Affinity is too high for a substrate and it can't dissociate from the enzyme?
For substrate. I mean, it could happen. You wouldn't want that to happen though. Because then like you can't dissociate and it can't make the interactions it needs to make like after that. So usually you want just the right amount of affinity for the substrate and then you want to be able to dissociate from that product once it's made. So you should have a lower affinity for a product than you do for its substrate. Yeah. Like a higher KD for the product. Yeah.
So I recall Dr. Francis mentioning that she might have a question for like list names of proteins and say which of these are enzymes. And so like, as far as I remember, like a general rule is like the ACE is an enzyme, but like, for example, rRNA would be considered an enzyme because that's a ribosome, right? Like that's what catalyzes the peptide bond.
Yes, generally, yeah. rRNA.
Well, is it an enzyme or is it just the catalytic component? Is it, like, if something has a catalytic component, does that make it an enzyme?
It doesn't necessarily make it an enzyme.
It doesn't necessarily?
No, because Stevia polymerase is an enzyme. Having a catalytic component doesn't make something an enzyme. It just. Right? Am I right?
Well, it doesn't necessarily make it a protein, but it makes it an enzyme. If it's speeding up a catalytic reaction.
What would be an
Take it back.
example of an enzyme that isn't a protein?
Ribosome.
A ribosome?
Well, the RNA component of the ribosome is the actual enzymatic.
So is that an enzyme though? Or is it a catalytic component?
The well, you call it the ribosome.
Yes,
It's
which
like.
is this.
For it.
If it has RNA material in it, does that immediately like make it impossible for the whole unit to be considered a protein then?
I wouldn't say it that abruptly, but I would just. I mean, it just goes down to saying like, there are non-protein catalytic, like, functionality of non-protein molecules. It doesn't have to be, enzymes don't have to be proteins, basically. They typically, but they don't have to.
How do you inject those cells? It's an enzyme.
Ask the question.
What?
Ask the question back. So what's the function of any enzyme?
Speed up reaction.
Yeah, so if it's catalyzing a reaction, then it's an enzyme.
And then the general like list rule is the ACE rule. Like if it ends in an ACE, it's probably an enzyme.
But it's weird because like DNA enzymes like build things, but then like every other enzyme breaks, then they think it's named after. So like don't go off of like naming rights.
I don't know if I ever really understood this, but, cause like, I remember her saying that RAN gap, even though it increases the reaction rate, it's not an enzyme.
It's because RAN itself is an enzyme, and by gap binding, it helps speed up its intrinsic rate of
Okay.
autolysis, yeah. So gap itself isn't an enzyme, it just helps RAN be a better enzyme.
Oh, okay, thank you.
Non-enzymatic, you said?
Non-enzymatic.
I think. I guess like GAP is something that's like speeds it up, but it isn't exactly an enzyme.
Oh, the G protein coupled receptor signaling molecule doesn't necessarily have to be a protein, and it acts as the GEF monologue for a GPCR. But that's tomorrow's review. But that's also another example of something that doesn't have to be a protein. Because there's a lot of signaling molecules that aren't proteins.
All right. So kind of how enzymes work. This is a little bit of a gen chem, but so enzymes generally work by lowering the activation energy or the transition state for that is required for a reaction to happen, which is somebody once told me it's the height of the hill. It's lowering the height of the hill, which is the activation energy. This is the only thing an enzyme is doing. It's not like changing the free energy of the substrate. It's just changing the energy required for the reaction to undergo. The reaction.
Do you know how enzymes are doing that, or just that they do that?
Trying to think about the questions. There was the question you had. I don't think you need to know like the mechanism by which they do it, just that it like lowers the activation energy. Yeah, and that's kind of the how.
I think I remember saying like on the plan that like the inside can put it is like position that makes it prone to forming the products. I said like the substrate binds and enzyme, the enzyme might like force it into a position that like makes the bonds. Okay, that makes sense.
Like a conformational change. And then this reaction. And endergonic or exergonic. Yeah, that's exergonic. You're releasing energy in the system. And then enzyme affinities. Usually enzymes and reactants should have high affinities, and then their products, you should have a lower affinity for products, just because you want the product to dissociate and do what it has to do in the cell. And then a good affinity with the reactant makes sure that the reactant isn't going to dissociate before the enzyme does its job and kind of yields product. That's it. Just know that your affinity for product should be lower than your affinity for substrate.
Do you have any examples where that would be the opposite? Like, is that something we would mention?
I don't think we've talked about anything about, maybe like in chemistry somewhere, but not here. Okay.
All right, so now we move on to topic 3, which is protein trafficking and intro to gene expression. Okay, so starting off, protein trafficking. Proteins must be trafficked to specific sites inside and outside of the cell in order to function properly. For example, insulin is a hormone that will be released into the bloodstream, and DNA polymerase has to stay in the nucleus because it's going to carry out DNA replication. In eukaryotic cells, we use signal sequences in order to localize these proteins. So to give you an idea, proteins that are going to stay in the cytosol, they will not have any signal sequence in their primary structure, but then for the endomembrane system, they will, but I'll get into that a little bit more. So for example, the endomembrane system, this includes the ER, Golgi body, lysosome, and cell membrane, and the transport vesicles that move in between these structures. If any protein has to enter the endomembrane system, it has to have an ER localization sequence. So if it has this ER localization sequence, which will be on the N terminus, it'll enter the ER first, and then that sequence is going to be cleaved once it's in there. Yes. Yeah. I think this is the first, yeah. This is the only slide that I've done so far. Got it? This is the only, okay.
Like, not backwards, like forward.
Oh,
There we go.
okay.
Oh, shoot, it was the same.
Yeah.
I got mixed up.
No worries.
I thought the sequences of it weren't in there.
Oh yeah, no, that makes sense. Okay, yeah, so any protein that functions in the endomembrane system, including membranes, it needs a ER retention sequence in addition to the ER localization sequence. So otherwise it'll be secreted from the cell. So if the primary structure of the amino acid that's being translated only has an ER localization sequence, it'll just make its way through the endomembrane system and then be secreted by the cell. So for example, insulin, a hormone, would be secreted from the cell, so it would only have an ER signal sequence. So getting more specific into signal sequences, they are a group of amino acids in an amino acid chain, so it's programmed right into that primary structure. And it's found in the initial DNA template as well. So that dictates the protein's destination.
And just to reiterate, it's not added after the proteins are made, no matter where it's found on the protein. It's in that initial sequence, and then certain proteins that have affinity for that sequence will recognize that it's meant to go to a specific place within the cell. So localization and retention sequences. In the endomembrane system, as I mentioned before, proteins must have an ER localization sequence. They'll first be sent to the endoplasmic reticulum and then enter the endomembrane system. And this will be at the N terminus, so at the very end of the polypeptide chain. And then in order to stay within the ER, Golgi, or the cell membrane, or lysosome, anywhere within the endomembrane system, they have to have that retention sequence that is also specific to the part of the endomembrane system that it wants to stay in. Then we have nuclear localization sequences. Those will be sent to the nucleus, and mitochondrial localization sequence will be sent to the mitochondria.
I'm gonna miss what you just said. For the retention sequences, did you say there's specific retention sequences for different areas of the endomembrane system, or?
Yes.
General one?
Yeah, exactly. So if a protein is going to the endomembrane system, it will always have the same, not necessarily the same, but it'll have the same function of having that ER localization sequence. But then if it's going to stay in the endomembrane system, that's where the retention sequence is going to vary, depending on whether it's going to, vesicles, staying in the ER, et cetera.
Is the N-terminus on the front of the amino acid chain?
Yes. Yeah. John, can you verify that? The N-terminus is like the consider the front.
Yes.
And then C is the back. Okay. Yes.
It's where it starts being.
I just wanted to double check.
Yeah.
Okay. Okay, so again, protein with an ER localization sequence, which is on the N-terminus, will be brought to the ER with the ribosome while it's being translated. So while the ribosome is translating the mRNA to a polypeptide sequence, it's transported to the ER and it'll start entering the ER. So that's called co-translational transport, because it's being transported while it's being translated. So once the protein starts to enter the ER, that N-terminal sequence, the ER localization sequence, will be cleaved. And from there, either the protein will continue all the way through the endomembrane system and be secreted, or if it has a retention sequence, it'll end up somewhere within the endomembrane system.
Are all localization sequences on the N-terminus?
No, so I'll get into this more, but it'll be on the N-terminus for the endomembrane system and the mitochondria, because the pores in the ER and in the mitochondria are too small for a fully folded protein to enter, so it has to go through kind of single file, one at a time, so it has to be on the N-terminus, and then that'll get cleaved off. Whereas, say, for nuclear import, the nuclear localization sequence is going to be just on the surface of the protein, which means it has to be coded somewhere in the middle of the polypeptide sequence, and that won't be cleaved off.
Is that N-terminus just one amino acid, or is it a sequence of a few?
It's a particular sequence. So we call it the actual N-terminus is the amino group, because on one amino acid you have an amino group and a carboxy group. So then when you make a polypeptide chain, there's going to be one end that has the amino group, and then at the very other end it's going to be the carboxyl group. So yeah, at the N-terminus you have the amino group, and then from there you have a specific chain that'll tell you.
The terminus itself is just that amino group or the carboxyl group?
Exactly. So that's just kind of giving you an idea of where on the polypeptide is this sequence, if that makes sense. Thank you.
When you mentioned that the retention sequence is the same with Golgi body and cell membrane, does that also imply that there is a Golgi body retention sequence and a cell membrane retention sequence?
So, so the retention sequence is different. I'm not sure I fully understand the question. But if I'm gonna try to explain. So the localization sequence is totally separate from the retention sequence. Every protein that's either being secreted or ends up in the endomembrane system anywhere will have an ER localization sequence. But in order to stay within the endomembrane system, it also has to have a retention sequence. So that's where you're gonna see the variation, if that makes sense.
What I'm saying is, do the Golgi bodies and cell membranes have their own specific retention?
Yes,
Gotcha, thank you.
Yeah.
That's what I thought.
any more questions?
Uh, how are they brought to the ER when they're being transitioned?
So the ribosome is gonna bind in the cytosol, right, and then start translating it. And then based on that localization sequence, the cell knows to kind of bring the translating, in the process of being translated protein to the ER. I don't know, I don't think she specifies what that mechanism is, but just know that if it has a specific localization sequence, the cell will know to bring it to that destination.
Would anything happen if the localization sequence didn't get moved on?
Yeah, so that's actually probably a good test question. If if you don't have any localization sequence, then it would just stay in the cytosol, right? Because proteins that stay in the cytosol don't have any retention sequence because the ribosome will just fully translate it in the cytosol and then it has nowhere to go, so it'll just stay there, basically.
What happens if once it enters, the ER, if the N-terminus isn't cleaved off?
That's a great question. In that case, I mean, it's already in the ER, so it could continue to its destination as long as it has the correct retention sequence. I don't think that.
Would that like affect folding?
Yeah, I mean,
Potentially,
if you're abiding by function as due to structure,
mhm.
if you're changing the structure, you're changing.
Yeah, because if that's a part of the process, then I'd say that if you leave that on, something unwanted will happen in general.
Don't some proteins that are, that have the ER localization signal sequence retain their N-terminal signal sequence when they're like transmembrane proteins?
I'm not sure about that.
I think in general, if it has like an N-terminus sequence to go into the endomembrane system and it's not cleaved off, those proteins are gonna be like retrotranslocated, they're gonna leave the ER. If that N-terminus sequence is kept, it's usually like leaving and it needs it to go back in. I think.
I'm sorry, I don't remember that detail. But that's a good question to write down and ask Laura, Dr. Francis, sorry.
Yeah, I guess just to like reiterate what we talked about, so like the reason why, for the ER localization, like why the N-terminus sequence is cleaved off, is that's just because like if it's kept, it like inhibits like the function of the protein for later work. Like if it, yeah, is that?
Yeah, I guess the way that I think about it is it's kind of served its purpose, doesn't need it anymore, so it can just like
Yeah.
take it off, yeah.
It's just like a shipping code.
Like a zip code, yeah, exactly. Yeah.
Thank you.
Yeah.
Is it possible if the N-terminus stayed for that protein to then try and be resent to the ER after being sent out or sent to a different area in the endomembrane system?
So once a protein enters like the mitochondria or the ER/endomembrane system, it stays there. So, because the localization sequence just makes it so that the protein will go to a specific place. So if it stays on, that doesn't mean it can leave, if that makes sense. So, yeah, it could affect the protein's ability to fold properly, but not necessarily its ability to like leave again.
Okay.
It won't add that capability. Any other questions?
So without a retention sequence, the end, like, destination for the protein would be outside the cell, right?
Yes, it'll be just excreted through like exocytosis.
Okay.
Oh wait. I don't know if I read all of this. Yeah, so that retention sequence will be on the C terminus, which is the opposite end from the N terminus. And then, yeah, so when it says same with Golgi body and cell membrane, that's just referring to the endomembrane, any part of the endomembrane system. It's not just those two specifically. That I'd say yes, because that's where the cell knows to recognize it. So if it's not in that spot, then it wouldn't. It would be looking for it at the C terminus, and if it's not there, then it would probably be secreted because it wouldn't be recognized.
And we don't need to know the mechanism by which the cell recognizes these sequences, right?
Just kind of understand the idea of affinity and amino acid interactions. So the way that proteins recognize the sequence is based on the specific charges or lack of charges on the amino acids in that sequence. And that gives them the ability to bind to certain proteins. So I don't have a specific slide for nuclear localization sequences, but we'll go over nuclear import and export. So here's just a summary of the different signal sequences. In the nucleus, the signal sequence will stay on the protein at all times. That's because it's encoded somewhere in the middle of the polypeptide, and it'll be kind of on the outside of the protein where other proteins can recognize it. So it will not be cleaved off because it's just right in the middle. Whereas for the endoplasmic reticulum and the mitochondria, those signal sequences will be on the N terminus, and they will be cleaved once they enter their destination, whether that's the ER or the mitochondria. And that cleaving is done by signal peptidase, which is an enzyme, which you can tell from the ASE at the end of the name. So just to talk a little bit about mitochondrial localization. So similar to the endoplasmic reticulum, the pores where the proteins enter is too small for a fully folded protein to go in. So it'll enter the mitochondria kind of in a single file fashion. And then once it gets into the mitochondria, it'll be folded. All right, so I mentioned that, but just to reiterate, for nuclear localization sequences, the signal is in the middle of the primary structure, so it ends up on the outside of the folded protein. For ER localization and mitochondrial localization, that signal sequence is found on the N terminus and will be cleaved. For nuclear localization, the protein will enter the nucleus fully folded and fully made. This is because those nuclear pores are large enough to fit a fully folded protein. So that's where you see a difference between nuclear import and ER or mitochondrial localization. The pore is just big enough in the nuclear membrane for fully folded proteins to enter. And that's why that signal sequence is found on the outside of the protein. So for both the ER and mitochondria, once the protein is fully entered, chaperone proteins inside the ER or mitochondria will help the protein to fold up. And finally, all right, I've said this a bunch, but in the bottom is just a reiteration of where you're going to find those signal sequences. Any questions on that? Okay, great. So here's a question for you. A protein is made to replace a receptor on the cell membrane. This protein was translated by a ribosome bound to the ER. Imagine that this protein underwent a mutation where the retention sequence was deleted. Assuming that the protein's ER localization sequence is still intact, where will the protein end up after being made?
If it loses the retention sequence, it would no longer be able to stay within the endomembrane system. It would be sent extracellular, like a hormone.
Yeah.
Because the retention sequence is what helps it stay down through the cell membrane, as that's a part of the endomembrane system.
Exactly. Perfect. Yeah. So here's the answer. The protein will end up being secreted outside of the cell. Remember, when proteins have an ER localization sequence, they are bound for the endomembrane system and will move all the way through the secretory pathway. In order to be stopped at any point along the endomembrane system, the protein will need a retention sequence. So going into the mechanism of nuclear import. So just to start off, to give you some context, RAN is an enzyme itself. It is a GTPase, which means it's an enzyme that can hydrolyze GTP. So you see that here. Its intrinsic enzymatic ability is to hydrolyze GTP to GDP, and then GAP is the protein that helps increase its intrinsic rate of hydrolysis. So for nuclear import, we start in the cytosol. We start in the cytosol. So we have a cargo protein that has its nuclear localization sequence that's going to be on the outside of the fully folded protein, as I mentioned before. So important is a protein that's going to recognize that sequence, bind to it, and then enter the nucleus through this nuclear pore complex. And then once it's inside the nucleus, RAN in its GDP bound state does not have affinity for important. So GEF, also known as guanine exchange factor, is going to assist RAN in exchanging GDP for GTP. When GEF binds to RAN, it decreases its affinity for GDP, which allows it to let go. And then GTP, which is in decent concentrations in the cell, can diffuse in. And then once GTP is bound to RAN, it gains affinity for important. I'm just gonna stop there and see if anyone has questions. Okay. So now that GTP is bound to RAN, it has affinity for important. When it binds to important, that causes important to lose affinity for its cargo, and then the cargo is released into the nucleus to do whatever it's supposed to do. So then from there, important has to be recycled back into the cytosol so that it can continue to import more proteins into the nucleus. So RAN GTP, still bound to important, is going to help important leave the nucleus through a nuclear pore. And then once in the cytosol, RAN GTP needs to let go of important, and GAP is going to help it do so. So remember that RAN GDP doesn't have good affinity for important. It's the same for whether it's in the cytosol or the nucleus. RAN GDP will have poor affinity for important. So when RAN hydrolyzes GTP to GDP, it will let go of that important. And RAN's intrinsic rate of hydrolysis of GTP is very slow. So in order to help it do so on a reasonable timescale, GAP will bind to RAN and assist it in increasing that intrinsic rate of hydrolysis so that RAN can become RAN GDP and let go of important so that it can continue the cycle all over again.
So with the intrinsic rate of hydrolysis being that low, is that why when it's in the nucleus, RAN does not hydrolyze the GTP before exporting?
Yeah, probably. I mean, I don't think she says that in class, but that's good reasoning for why that would be the case.
And with that, is there no gap present inside of the nucleus?
No, so GEF is localized to the nucleus specifically, and GAP will be localized to the cytosol.
So just to be clear, RAN is the enzyme that like performs the hydrolysis. But then GAP and GEF just like help facilitate RAN. So,
Exactly.
Wait, what is it classified as again?
So the way that their official kind of definition are just proteins that help help enzymes in the RAS family to carry out their function, and that definition is going to be on one of these slides too.
Okay.
But yeah.
Okay, thank you.
And GEF, just sorry. GEF just makes a conformational change to RAND that increases affinity or decreases affinity for GDP.
Did you say GEF?
Yeah.
Yeah, exactly. So when GEF binds to RAND GDP, loses affinity for GDP, and then that'll diffuse out, and then GDP can diffuse in.
And it doesn't stay bound to RAND, right? It just causes a conformational change and then releases it.
Yeah, exactly. So, what can help you kind of remember that is GEF is wanting exchange. So think of GDP being exchanged for GTP. It's not adding a phosphate, it's just fully exchanging those two different molecules. Whereas GAP is, I think, wanting or something just think activating. So it's helping to activate RAND's intrinsic ability to hydrolyze GTP. Any other questions on that?
I mean, I'm going back to like a question that was on our first exam, which asked about, if RAND's affinity for GDP was decreased such that GDP diffused out, and how that would impact this cycle. And so, and I got that wrong, so I'm just, kind of trying to reinforce my understanding, which is, so looking at this, the GDP in the cytosol would diffuse out and GDP would bind, which means that RAND GDP would stay bound or like keep binding important in the cytosol, which is why the rate of import would decrease.
So the original question was saying that it loses affinity for GDP. I'm
Yes.
just clarifying.
Yeah,
Okay,
a mutation that decreases the affinity of RAND for GDP such that GDP dissociates out of the RAND binding pocket as soon as it binds.
okay, yeah, so in that case, GTP is just kind of around in the cell. So if GDP isn't bound to RAND, then GTP is likely to diffuse in and bind to RAND. So then that means it's kind of in its active state. And in its active state, it's bound to important. But if you look in the nucleus, when RAN is in its active state and it binds to important, it causes it to let go of its cargo.
Yeah.
So the important part of import in the cytosol is that important can bind and stay bound to the cargo. So if we had active RAN in the cytosol, it would bind to this complex and cause it to let go of the cargo.
Okay.
So then that just kind of disrupts the whole process.
Okay, thank you.
With that question, why would it disrupt the cargo if GAP is still present? Because wouldn't the GTP that binds to RAN just get hydrolyzed by GAP and then still prevent RAN from being activated?
So to a degree, but in general, kind of if everything else in the cell stays the same, like the different concentrations of everything, I guess the way I think about it is
Would there be like more important than RAN would bind to? Or?
that's what I say. Like it's not prepared for the amount of RAN GTP that there would be. Like certainly GAP would still be present and be able to help hydrolyze GTP to GDP, but it would once it hydrolyzes GTP to GDP, if it doesn't have affinity for that, it'll just diffuse right out and then GTP would come back in.
Okay.
So it would be kind of a competing process, but the bad process when we don't want would kind of win and disrupt the whole process. Any other questions on import for now? Okay, so going through nuclear export, which uses pretty similar and a lot of the same proteins. So we're starting in the nucleus. So we have a cargo protein, which has a nuclear export sequence on the outside of the protein. So exportin on its own has to be bound to RAN GTP in order to have affinity for the cargo protein. So same process as an import. RAN GDP will be helped by GEF to exchange that GDP for GTP. that kind of activates RAN, and then it can bind to exportin, and then exportin now has affinity for, the cargo protein. And then this whole complex will exit through the nuclear pore. And then once in the cytosol, in order to let go of the exportin, RAN helped by GAP will hydrolyze the GTP to GDP. That causes RAN to lose affinity for exportin, and since exportin needs to be bound to RAN GTP in order to have affinity for cargo, that'll cause these two structures to separate as well. And then the way that then exportin can just re-enter the nucleus.
I'm sorry, does RAN bind to the nuclear sequence first before it binds to exportin, or is it? Sorry, is the exportin the MES first, or is it RAN to exportin?
It's RAN, exportin, then exportin, NES.
How does exportin get back into the nucleus? Is it through an important, like?
It can just diffuse through.
It can
just diffuse those crystals?
Yeah, it diffuses through the pore. It doesn't need it.
And other things do that?
Yeah, I don't think for this class,
I'd say in general, yes, but you don't need to know examples, or you don't need to know that.
But so essentially for this class, if it's in the nucleus, it has a nucleus insert sequence.
Yes.
Unless it's.
Yeah.
I think. Well, exportin and importin both need to be able to cycle through. So they both need to have a nuclear import and export, because they have to both enter the nucleus and leave it. So any protein that's going into the nucleus needs to have a nuclear localization sequence, and any protein that's exiting the nucleus has to have a nuclear export sequence.
Okay.
In both of the import and export paths, RAN is only ever leaving the nucleus, so where is the RAN coming from in the nucleus? Like, would there not be like a disproportionately large amount of RAN in the cytosol then?
There is a way, so RAN does get cycled back into the nucleus, but we don't go over that in this
Okay,
class.
so we just assume that it's cycled through.
Exactly.
Does the phosphate group do anything specific?
This is just to show you, that when, RAN hydrolyzes GTP to GDP, the way that that happens is GTP is losing a phosphate group. So then GTP goes from triphosphate to diphosphate, and then you just have this extra phosphate.
the I means inorganic.
Yeah. Okay. Any other questions on export?
Oh, actually, yeah.
Yeah.
So since RAN GTP is needed, inside the nucleus for both import and export, is there any competition between the two where like export could be inhibiting the amount of important that could be released once it's entered the nucleus, or is there just enough for both processes to occur simultaneously?
Yeah, I'd say that both processes, pretty much every process in the cell is regulated pretty tightly, so we don't talk about the specifics, but the cell kind of makes sure that whatever factors need to be balanced in order for these processes to take place the way they're supposed to, kind of take care of that. Yeah.
And then generally speaking, the concentration of GTP is much higher than GDP in the cell in general. Okay, so then it allows for, because the necessity for energy releasing molecules is great for, it doesn't matter if it's ATP or GTP. The concentration of the activating protein, or I guess it's not protein, the concentration of GTP in the cell is generally higher.
Does RAN have a naturally higher affinity for GDP?
GDP?
Yeah.
Well, they each have their own like affinities for specific proteins. There isn't like a way of determining that.
Okay.
But while bound to GDP, it remains an active.
Because I'm just curious with like, when RAN's in the cytosol, what's preventing RAN from? Since there is obviously a large amount of RAN, what's preventing RAN from just being very populous in the GTP bound state, like in the cytosol where that would be?
Because RAN's an enzyme. RAN's a GTP, the function of a GTP, or RAN's a GTPase. The function of a GTPase is to hydrolyze that bond, right?
Okay, so I guess it's always trying to continue that function.
Well, it has other functions that it performs within the cell, but the intrinsic enzymatic activity of a G protein is to hydrolyze that phosphate.
Okay.
What I'd also add on to that is that the cell really carefully, again, regulates how much it's making of everything. To kind of make sure, because like if concentrations are out of whack, then certain cellular processes aren't going to work properly. So that's the best way that I would explain that. If you're ever wondering like about different concentrations, the cell tries to
Yeah,
Yeah.
I just wanted to make sure there was no like difference in affinity for GDP versus GTP.
not that you need to
Okay.
know of. Yeah, just know like what the difference is when it's GTP bound versus
Right.
GDP bound.
Okay.
So talking more specifically about GEFs and GAPs, the general definition is they're terms for proteins that act on the RAS family of GTPases. And this includes RAN. So again, GTPases are enzymes that are able to hydrolyze GTP into GDP. So GEF stands for guanine nucleotide exchange factor, and these facilitate the exchange of GDP for GTP. GEFs are found in the nucleus. And the way it works is GEF binds to its GTPase, such as RAN, which causes a conformational change, which causes RAN to lose affinity for GDP, and then GTP is just around and can diffuse in, which puts RAN in its quote unquote activated state. So then GAPs are GTPase activating proteins. So these are found in the cytosol, and they stimulate the intrinsic GTPase activity of RAN. The way this works is GAP binds to the GTPase and sticks an arginine into the active site of the GTPase. And this kind of alters the chemistry of that active site such that it increases the rate of the reaction. So RAN is able to hydrolyze GTP to GDP at a quicker rate than it would be normally.
If you could go back, like,
Yeah.
whichever slide had the import chart, like the diagram, there's the GTP that's going through RAN gap. Is that GTP the GTP that's binding to RAN, or is that a GTP whose energy is used for the process?
That's a good question. For like up here.
Yeah, like you see how like the GTP, the arrow is
Yeah.
going through GEF, is that like the energy that's used for GEF to bind to RAN, or is that just the GTP that ends up binding?
So I'm just gonna start by saying definitely, all you need to know is that GEF helps exchange GDP for GTP. I'm honestly not sure whether that shows a coupled reaction, or if it's.
No, that's just indicating that the RAN with the help of a GEF is going from GDP back
Okay.
to GTP.
Okay.
Okay, yeah. Good question though. Okay, and just to reiterate, GEF and GAP are not enzymes, they're regulators of enzymes, and that enzyme is RAN. All right, any questions here? Okay. So here's another question for you. How would nuclear export be affected if there was a loss of function mutation in RAN GAP? So that's, and we're looking at export specifically.
If there was a lot of function in RAN gap, that means that the function would never be able to release the cytosol because gap takes the GDP and turns into GDP. I don't know the reason why I'm using these phosphate.
Yeah, so what that's showing is that the GTP is losing a phosphate.
Gotcha.
And so then you end up with GDP and a phosphate. Because if you added this phosphate back onto GDP, you get GTP.
Yeah, but my primary point is
Yeah.
that if that was to have a loss of function, then the cargo would never be able to make sense.
Because GAP's function is to increase the rate of hydrolysis, and if the hydrolysis is happening really slowly, then it's not gonna unbind from exportin.
I have a question about that. Is it important, the fact that exportin wouldn't also be released? So would that decrease levels of export? Because exportin wouldn't be cycled back into the nucleus?
Yeah, you could also,
Not big.
no, you could also say that, yeah, because yeah, the complex will all stay together. And there's still more, it's not like an isolated situation, there's still more exportin in the cytosol, but if all gaps lose their function, then you will get kind of a flaw in the cytosol.
Like, for GTP, just like hydrolyze GTP, like eventually. Like,
Yeah, it would happen eventually.
So like, if I answer the question by saying, oh, like, the process of nuclear export would just be like severely like slowed down and like, disrupted. But like, just like slowed down, would that still be right?
I'd say the most important thing is explaining what it would mean for RANGAP to have a loss of function, like it would be important to explicitly say if GAP isn't functioning properly, then it's not helping RAN hydrolyze GTP. So that process is happening at its intrinsic rate, which we know is extremely slow. So just like kind of stepping through the checking all your boxes, but yeah. You're welcome. Okay, so I'll just read the answer. RAN gap is responsible for increasing the rate of RAN's intrinsic catalytic ability. If RAN gap did not function correctly, GTP will take a long time to be hydrolyzed into GDP. This means that RAN GTP would remain bound to exportin and the cargo protein would not be released. Over time, nuclear export will stop as all the exportin proteins accumulate in the cytosol and are unable to re-enter the nucleus. Again, for this answer, I'd be careful with using the word all, but there would be a level of clogging the process. Okay, so getting into transcriptional regulation. Transcription factors, sorry, before I get onto this, are there any more questions on protein trafficking? Okay, great. So transcription factors regulate DNA polymerase binding to promoter DNA. These are basal transcription factors in eukaryotes and sigma factors in prokaryotes. And a positive regulator will upregulate transcription, so you'll see more transcription, and a negative regulator is an inhibitor, and that'll suppress transcription. So one of the ways transcription is regulated is how tightly chromosomes are packed, or DNA is packed. So euchromatin is the open form of DNA. So that would be this one. So that DNA polymerase can actually access the DNA. And heterochromatin is where the DNA and histone complexes are really tightly wound and packaged together, like here. So then DNA polymerase can't really access the DNA to transcribe it. Yeah. Okay, so RNA processing in eukaryotes. These are both ways of helping prevent the RNA from being degraded. So the 5' GCAP is a covalently added guanosine on the 5' end. So this is not encoded in the original amino acid sequence. It's added on after it's been made. So this prevents RNA nucleases from chewing the 5' end of the mRNA, because that's their function. RNA nucleases chew RNA. And it's added in the opposite direction. So poly A tail is on the 3' end of the mRNA. And this doesn't really prevent the chewing, but it kind of creates a buffer so that if a nuclease is chewing the end of the mRNA, it's not getting to the actual coding material. And then the poly A tail is also required for export from the nucleus. So you can think of it as a kind of nuclear export sequence. I didn't mention this, but I want to, for the 5' G cap, it's different from the poly A tail because the way it functions is it makes it so that it's too big for the nuclease to kind of fit it. So it can't get to the mRNA. Yeah.
Nucleases choose both directions, like 5 to 3 and 3 to 5?
Yes, yeah, so these specifically would be exonucleases, which means they chew at the end. And there's also endonucleases that'll chew in the middle, but this is specifically protection from exonucleases.
And this would be like during transcription?
So RNA processing takes place after transcription. Yeah.
So this would be like a post-transcriptional modification?
Yeah, exactly. Any other questions on this? Okay, so going into splicing, introns must be spliced out before translation can occur. So introns are parts of the mRNA that will not code for a protein ever. So they're just literally going to be cut out and removed. Exons are areas that are transcribed and translated. So where introns are going to be removed, the exons remain. And the spliceosome is the enzyme that cuts out those introns. Alternative splicing is a process that allows for the cell to be choosy about which exons are actually translated. So this allows for multiple different proteins to be made from the same stretch of DNA. So as this little diagram shows, there's a difference in between these two mRNAs. This one has a yellow section here, whereas this one kept the green one instead of yellow. So that would be an example of alternatively splicing exons.
And that would happen after introns are spliced?
Yes.
Do we need to know the mechanism by which the exons are then connected after introns are removed?
I don't believe so. Yeah. Okay, yeah. So basically takeaway from that is not all exons are necessarily going to be translated, but none of the introns ever will.
And this is still a post-transcriptional modification, correct?
Yes. Okay, so here's kind of an overview of all the different ways, or some of the different ways gene expression is regulated in eukaryotes at different levels. So we have transcriptional control. And an example of that would be your euchromatin versus heterochromatin. So open versus closed conformation of chromatin that allows for DNA polymerase to either access or not access the DNA. Then you also have transcription factors, which are required to bind in order for polymerase to bind. Then you have processing control, which includes RNA splicing, so taking out introns, and then also alternative splicing. And then that would also include the 5' cap and the poly A tail. Transport and localization control. This would include sending the RNA to the P body, which is kind of like a closet. The cell will send it there for storage or for degradation. We don't really know what goes on in the P body though. So this is happening in the site. All this happens in the nucleus and then here in the cytosol. So degradation control, that would be your, think like RNAi, RNA interference, siRNA mediated mRNA cleavage and degradation. Yeah, that's an example for that one. Translation control, which includes just totally blocking the ribosome binding to the mRNA. If the ribosome can't get to the mRNA, it can't translate it to a protein. For protein degradation, an example of that would be ubiquitin-mediated proteolysis. If the protein isn't folding properly or isn't functioning properly, it can be degraded. And then protein activity control. As we know, you can control proteins by phosphorylation, dephosphorylation, binding to different proteins, like you have cyclin CDK complexes that influences the activity of CDK. And then you could also include, say, GTP binding and hydrolysis as a way to control protein activity.
So of these, which would be considered the post-translational modification?
Okay, post-translational, that would be like anywhere after you see mRNA. Or, no, I lied. That would be anywhere after you see protein, sorry.
Okay.
Yeah. Any other questions on that?
Going back to the slide that had the view chromosomes and heterochromosomes.
Yeah. We'll also send out these slides, but yeah. Okay, and then going into differences between eukaryotes and prokaryotes when it comes to regulation. So both types of organisms regulate mRNA degradation rates. They regulate mRNA localization. They regulate the rate of translation, protein activity and degradation, as well as transcription using transcription factors. And differences, eukaryotes package DNA in a chromatin, which helps to control the ability for proteins to access the DNA, whereas prokaryotes do not package DNA into chromatin because they don't have histones, so their DNA just kind of sits around like spaghetti. Eukaryotes have a nucleus, so mRNA is processed and transported out of the nucleus, whereas prokaryotes don't have a nucleus, so there's no eukaryotic RNA processing for nuclear export. It's totally unnecessary. They do process their RNA, but you don't need to know how. And then they also differ in their main type of general transcription factor. Eukaryotes have basal transcription factors and prokaryotes have sigma factors.
Do prokaryotes have a pre-transcriptional regulation besides transcriptional factors, like how eukaryotes have the packaging, or is it just the transcription factors that we know?
I'd say, just since we focus mainly on eukaryotes for this section, just focus on, the difference in, and knowing that since there's no nucleus, it's not going to be exported, things like that. Yes, and assuming everything went well with the recording, that'll be on YouTube too.
Thank you.
Thank you. Thanks for coming, everyone. If you have any questions, oh, sorry.
We have, that's a funny thing. So, yeah, stop the recording.
Okay. How do I do that?
Is it stop?
Here.